Thank you very much, and it's great to be here to give you a bit of a snapshot about some of the innovations that may be coming soon to accelerate the rates of progress in plant breeding. And there's so many innovations, and sometimes we get caught up in this discussion about GM and gene editing, and we need to stop and reflect on what we're actually trying to do. And what science does is try to seek solutions to problems. And I think we all see the results of this in our daily lives. So if we think of the smartphone, this is a tremendous scientific innovation that scaled down from these computers the size of a room, which process data very slowly, into something that you can hold in the palm of your hand. Plant breeding is very similar to this. So in the 1960s and 1970s, Norman Borlaug took the traditional long-strawed varieties of the time and crossed them together with novel sources of reduced height. And these came from Japan, and he had the idea that if he made crosses between these tall and short varieties, he would produce material which had a better stature, you were able to apply nitrogen to. And this resulted in the semi-dwarfing types that are now grown almost all around the world in wheat production. And nature is possibly the greatest innovator. So I guess some of you may know that all sweet potato, no matter who grows it, is genetically engineered. So this is a naturally genetically engineered product. And the reason all sweet potato is genetically engineered is that during its evolution and domestication, it was infected by a bacteria called agrobacterium, which is the bacteria we use in the lab to create genetically modified plants. So what happened in sweet potato evolution is that it was infected by this bacteria. The bacteria entered into its genome, and this conferred a fitness advantage, so something that made the sweet potato even better. So this was a continued part of its evolution. So all of the material we propagate today contains this remnants of a natural genetic modification process. So we seek to find solutions. And I think there's no questioning the number of complex challenges which face UK and global agriculture. And in the arable sector where we work on cereals specifically, there is just this huge body of interacting complex challenges. We need to produce more yields and a higher quality of food. We need to do this in the face of chemistries that are being lost, in terms of new races of pathogens which are really fighting against our best efforts to breed new varieties. So we face an unparalleled and unprecedented set of challenges, and I don't think anyone would deny that these are extremely complex and interacting. And I always come back to this quote from Norman Borlaug, who produced these semi-dwarf wheat varieties in the 1960s and 70s. And that's really that science and the tools and technologies that we have at our disposal allow us the means to address these challenges. And so the work that we do really looks at how we can deploy these innovations, these tools and technologies to really drive progress in plant breeding. And today I'm just going to talk very briefly about 4 of the areas that we at NIAB are working on to address some of these challenges. And the first is using native traits, so going to the ancestors of wheat to try and find novel things that we can incorporate to support breeding progress. The second is the use of genomic information, so we have access to unparalleled amounts of data, and we need to exploit these for breeding gain. The third is the use of new breeding tools, and we can't underestimate the importance of these tools in understanding what we're doing as part of change in the biological process. And then finally, characterization at scale, how we understand these things in the field. And as researchers, it's very important for us to translate this knowledge into practice. And my key message is that elements of all of these approaches can be incorporated to address the key challenges facing the industry. And just for some context, this slide was provided by Jack Watts at the NFU and outlines the nitrogen challenge, which is one that we're working on quite intensively. So although we have increasing wheat prices, we have increasing costs. So ammonium nitrate costs are increasing steadily. And we also have the emissions side of this, both in the UK and elsewhere in the world. So this nitrogen challenge in wheat production is very pressing, and it's something we can address through our research. So we do this in a number of ways, and I'll talk about these over the next 4 slides. And the first is to look at native trait variation. So this is the variation that exists in nature. So wheat is hugely genetically complex. Its genome, the genetic information it carries, is 5 times the size of the human genome. So this is a huge, intractable beast that we need to work with. And the reason wheat's genome is so complex is that it actually combines in its genomes the genomes of 3 ancestral grass species. And these grass species came together during the evolution and domestication of wheat over 2 successive events. The first event brought together the original 2 genomes and produced something called Triticum dicocoides, which has been domesticated to pasta wheat, the wheat that we use to make pasta and semolina products. And then much more recently, about 10,000 years ago, a 3rd genome joined the party, and this is called goat grass, Agelops tauschii. And we're really interested in this wild ancestor of wheat, and we're really interested in it because we know a lot of the things we want to change reside on this D genome contributed by this unique grass species. We also know that the grass species is hugely diverse, so if we look at the genetic information, we see there's a huge distribution of genetic material. And then if we look at the geography on top of that, we can see that its native range is immense. So it covers a wide ecogeographical range, and we could hypothesize that in this range of environments, novel plant types are required for foraging for nitrogen in low nutrient soils, for disease resistance, etc. So over a very painstaking number of years, we have tried to capture the variation in these wild relatives of wheat in this goat grass ancestor. And we've done this through 10 years of intricate hand pollination, so crossing together the different species to resynthesize or recreate these evolutionary events in wheat's domestication process. And then we move from that into elite wheat varieties. And our recent results show that we now have material from this program which goes back 10,000 years to the start of this domestication evolution process that out-yields our commercial controls. So this is really promising. We've gone back 10,000 years. It's taken us 10 years to do that, to try and mobilize some of that novel variation which is hidden in the wild relatives of one of our key crop species. So when we do that process, when we create this new diversity, we go back to our native traits in the ancestors, in the gene pool of wheat. And the analogy there is that we're building a haystack of material. We're trying to capture all of this new material, and then we're trying to search for a needle in that haystack. But we can take a much more targeted approach, which is what I'll talk about now, about the use of genetic information. So we know in model species that we can differentiate plant types into 2 fundamental differences, which we call James Dean and George Clooney. So one, live fast and die young. So this has no differential response, no matter how much nitrogen you put on. Whereas we have a second plant type which waits around, hoping George Clooney may one day show up. So this is a plant type which responds very rapidly to the application of nitrogen. So we can think about this finding in model plant species and look at some of the genes that we think in wheat might control that response. And then we can target the specific genes that we identify to modify leaf angle, branching of tillers, and the remobilization of nitrogen from the leaves into the grain. And when we do this, we use genetic modification because this is a rapid way for us to very precisely target a gene of interest. So you will have heard a lot about gene editing and CRISPR tools. And CRISPR tools are essentially a molecular pair of scissors where you're cutting a section of the genome, and then because the DNA can't put itself back together, it stops the function of that gene, so it knocks it out. But what we're actually doing in this experiment is asking using the genetic modification as a tool to supercharge a gene that we know is already present in the wheat genome. So we want this gene to be switched on higher. We want to put the pedal to the metal, and we want it to maximize the uptake and use of nitrogen by the plant. So this is called an overexpression line. So we're overexpressing, ask the gene to produce more copies of the protein. And our early results from this work are very exciting. So when we do this, when we turbocharge specific genes, which we hypothesize control the nitrogen response, we're able to promote the use of nitrogen within that plant and to convert it into yield. So this is really exciting use of GM tools to enable us to supercharge genes that are already present in that plant. And then finally, we need to be able to scale things up into the field. So obviously, we're not in a good place to do that with GM or gene-edited products, but we are able to use these kind of systems. So this is an example in collaboration of ADAS of using OptiPlot trials, so 6 rates of nitrogen on very small amounts of seed that allow us to compare some of our native trait material. So this is material that's coming through our pipeline of discovery using the ancestors of wheat and compare its response to nitrogen to elite varieties. And this allows us to identify promising material early on that confers a nitrogen advantage. So just to finish, I've covered a few different areas where innovations may come to plant breeding. I think it's very important that we focus on the challenges. And if we come back to the analogy of the iPhone or the smartphone, there are 12 separate scientific breakthroughs which made it possible for that to come together. And I think in plant breeding, we're in a similar situation, that aspects of all of these innovations will come together to address the challenges and deliver impact to the industry. Thank you.
Thanks, Alison.